Grace, who's a senior. She's never presented at Journal Club before, but she's going to get treated as a third-time presenter today, which means that you can just pelt her with questions. She's presenting on a new link between two parts of the brain and fear and how that's related to PTSD.
All right, hi everyone. Happy what? All right, hi everyone. Happy Thursday. So my paper is titled Integration of Projections from the Basolateral Amygdala to the Entorhinal Cortex Disrupt the Acquisition of Contextual Fear. Or in other words, how blocking certain neuronal pathways can decrease fear. So we all heard of PTSD. Post-traumatic stress disorder elicits like anxiety-like symptoms and occurs after seeing, hearing about, or especially experiencing a trauma of some kind. So whether that's war, assault, rape, abuse, etc. All these things can cause PTSD. PTSD is actually a big problem. It affects about 7 to 8% of the adult population. So it's definitely something we need to be looking more into. And finally, it's based on contextual, as opposed to your normal cue-based fear. What is contextual fear? Contextual fear is best explained by example. So let's say you're 5 years old, you're in your room, and all of a sudden it starts to storm. Now lightning doesn't bother you, but thunder, like the noise it makes, the way it shakes the house, terrifies you. Since you're a smart little 5-year-old who's going to go to Pingree someday, you realize that every single time it lightnings, thunder immediately follows. Over time, you become scared when you see lightning because you associate it with thunder. This is contextual fear, becoming scared in environments or situations that remind you of a trauma of some kind. So, like I said, it's involved in a lot of anxiety-based problems, in particular PTSD. Okay. This is why we all hate computers. Okay, so some terms to know for the basolateral amygdala. The basolateral amygdala is part of the brain, and it regulates emotions, in particularly fear. The entorhinal cortex is another part of the brain, specifically right near the hippocampus, and it regulates processing context and images. Glutamatergic neurons. Okay, so a neuron is a brain cell. There's an example of one right here. And all I really need you to understand about glutamatergic neurons is that it's a certain kind of neuron, and we can separate it from other neurons that deal with other neurotransmitters and other pathways. For the purposes of this paper, that's all you need to know. Alright, so acquisition of contextual fear is that moment when you associate the trauma in a context. So it was when 5-year-old you realized or associated the fear you felt from thunder with lightning. Whereas expression of contextual fear is when you're re-exposed to the context without the trauma. It would be like seeing lightning by itself, but without thunder, and still being afraid. You've already acquired that fear. You already associated thunder and lightning. Is everyone clear on the differences between those two? Alright, good. So, this is a cross-sectional diagram of the brain. Just to give you guys a visual, this is the amygdala right here, this little ball-shaped thing. And just beneath the hippocampus is the entorhinal cortex. These are the two areas of the brain we're focusing on today. So just to give you an idea of where everything is located. Background. Going into this experiment, the researchers knew that the basolateral amygdala and the hippocampus were involved in contextual fear, somehow, some way. What they didn't know was whether there was a direct connection between the basolateral amygdala and the entorhinal cortex. They also didn't know if it existed, whether it regulated acquisition of fear or expression, or maybe both. we know that, what we don't know is whether specifically the basolateral amygdala, the entorhinal cortex, are directly connected. Whether there's a projection from the BLA to the EC.
So literally connected?
Yes, literally connected. And if there is that connection, we don't know what it regulates.
Okay, so there's two parts of the brain.
Yeah.
Important in the actus, well, don't know what.
Yeah. Important in contextual fear, don't know what part of contextual fear.
Right, so okay, so we know that two parts are responsible for kind of this generation of fear, but we don't know whether it's acquiring that fear or it's in the recall of that fear.
Yes.
Okay, and we want to see if those two parts are connected.
Yes. Awesome. Everyone good? Yeah? Alright. So to test this, the researchers use something called optogenetics. So if you're up on your classical roots, and that's opto, we're talking about light and DNA here. So optogenetics is actually pretty simple on a broad kind of view. What we're doing is we're taking DNA originally from algae, and this DNA codes for something called a light-gated ion channel protein, right here. So ion channel protein part first. Ion channel proteins are transmembrane proteins. They sit in the middle of the cell membrane, and they control concentration of ions in and out of the cell. Specifically, in our case, in and out of a neuron. Light-gated just means light controls this particular ion channel protein. So why this is so important in neurons specifically is that basically your brain works by neurons firing. And getting a neuron to fire is a very complicated event, but one of the things that goes into it is the balance, the change in the concentration of ions in and out of the neuron. So broadly speaking, if you can control the ions coming in and out of the neuron, you can control whether the neuron will fire. If light controls that protein, we can use light to control whether or not the neuron fires. That's, broadly speaking, that's what optogenetics does. Researchers use it to control certain neurons that they make express this protein, which I'm going to call an actuator from now on. It's what is called an optogenetics, and it's a lot easier to say. Any questions? Now would be a great time to ask them, because optogenetics and contextual fear are the basis of this presentation. Anyone? The experiments. So, the researchers took a virus, AAV. They took the DNA that codes for those actuators I was just talking about, the ones that we can express and use them to control the neurons and light. The DNA for those go into that virus, which had into something called EYFP, which is this thing right here. It's an fluorescent protein. Basically, it'll glow when it's expressed. So if we fuse it to the actuator, whenever the actuator is expressed, it'll glow. The area will glow. Just so we can see where it is. This part's the actuator. I'm not going to say the names, they're way too long. But basically, those are the actuators. They're under control of a certain promoter, CAMK2 alpha, and we injected them directly into the basolateral amygdala of the mice. Specifically, we targeted expression to the glutamatergic neurons of the mice. So remember how I said that's all you can know, that it's a certain kind of neuron? What they did was they took a certain kind of neuron, expressed the actuator in those neurons, so now we can control those neurons with light. They did this for every single experiment I'm about to explain, so just keep that in mind as we're going. And yeah, here's the basolateral amygdala, little nice needles. Slice this out.
So they did the transformation in single cell, yeah? The transformation they did in single cell, right? And then it grew into the whole organism, right? So it's just being turned on in certain cells.
Yes.
Okay.
It's turned on in certain cells.
But they did it in a single cell. They did a transformation in an embryo.
They injected it, yes, in a virus. Because of the certain promoter they use, that's why it's only expressing in certain cells.
But what I think he's asking is he's asking whether the virus was injected into a certain part of the brain,
Yes.
or whether all glutamatergic neurons are.
Oh, all right. I misunderstood your question. Thank you. No, it's only the glutamatergic neurons in the basal
actually injected it into an adult,
Yes.
not an embryo.
And microinjection needles, I developed the BLA, and they injected it into the BLA.
Okay, cool.
Yes?
So I just want to clarify. So when they target the expression of glutamatergic
Yeah, that's
matergic neurons, this causes people to have less contextual fear.
No, not. I'll go into what happens in fear. Basically, this is what they're doing for every experiment. This allows them to control the glutamatergic neurons with light. So by shining different wavelengths of light, they'll control whether or not the neuron fires or not. So it will give me another slide or two, and if you're still lost, ask another question. I think it'll make sense after I explain the next slide. Yeah, Andrew?
What kind of light does the activator react to? You said very low yellow, so I assume that wouldn't activate the activator, right?
For inhibition, they use the 532 nanometer green light. For stimulation, I would 400 something, 450 nanometer light. Awesome. Yeah, alright, we gotta go. Okay. So, slice physiology. They use light to photostimulate the BLA glutamatergic neurons, so those are firing. It is a very complicated process to find out that in 49 out of 56 EC neurons, the BLA neurons have fired there. So here's the BLA is glowing because the activator is expressed there, tied to the glowing protein, remember? It should only be glowing here. It's the only reason where we inject it. But you see down here, the entorhinal cortex is also glowing. The only way that would be glowing is if there was a direct connection between the BLA and the EC. The EC did not get any glowing protein, so there is a connection between the BLA and the EC. Lovely. Now we move on to behavioral experiments. Once again, we're injecting the mice with the activator attacking the glowing protein in the BLA. Same as before. But since these are live mice now, we need a way to train them to be afraid. It is a standard fear conditioning tumor, like this one right here. Basically, you can shock them from the floor, and there is that 532 nanometer laser for inhibition. Alright. So, experiment set number 1 was to inhibit the BLA EC during acquisition. So, day number 1, have a bunch of mice, you put them in a box, you shock the mice. This makes the mice afraid. So they're acquiring contextual fear. But when the researchers did this, they used light to photo-inhibit the BLA-EC pathway. So the BLA-EC pathway is not working right now when the mice are acquiring this fear. Okay. So a day later, the mice go back in the box, no shock this time, because we're expressing the contextual fear, and also no inhibition. The researchers left the pathway alone. Excuse me. A week later, they go back in the box, no shock, no inhibition. Okay? So, that was the first set. The second set was to inhibit the BLA-EC pathway during expression. So day 1, mice go in the box, mice are shocked, but there is no inhibition, because we're not inhibiting the pathway during acquisition. Day 2, back in the box, no shock, but we do inhibit the pathway. A week later, they go back in the box, no shock, and no inhibition. Alright, it's in the first pathway, the first experiment, we knocked out the pathway when the mice were getting the fear. Second one, we knocked out the pathway when the mice were being recalling the fear. Studio, are you okay? Okay. Alright, so what did they find? They found that blocking the pathway, yes. Sorry, can you say that louder? They use light. Remember how I said we can control those neurons now? We can also make sure they don't fire. Okay? Alright, lovely. So, inhibiting the pathway during acquisition does decrease fear. Now, they quantify fear by something called freezing bouts. So as you can see, in this diagram, this is the experimental group where they inhibited fear during acquisition. And this is like normal mice. They'll do the same thing, but with no blocking of the pathway. These mice had a lot more of the longer duration crazy bouts. They were more afraid than the mice that had their pathway blocked during acquisition. So, inhibition during acquisition decreases fear. Inhibition during expression, however, does not decrease fear. When they left the pathway alone during acquisition and blocked it when the mice were re-exposed to contact, the mice were just as afraid they wouldn't be normal. Is any questions on this point? Yes.
So what day does that graph describe? Because they're 2 days after the experience. Well, you know how the first day they inhibit. So is this like the 8th day?
Okay, this is like a combination of the 2nd day and the 8th day, I believe, because of both day 2 and day 8 were both expression days. The reason why they did it a week later was because they wanted to make sure the effects lasted, which they did. So this is data on how scared the mice were on day 2 and day 8 when they were re-exposed to the box in which they had been shocked before. Does that make sense?
Yeah. Then, do you know which day there were more like reasons? Like, for example, if you inhibited, like, for the second experiment, they tried to inhibit, like, the expression, so.
Yes, it was day 2, but there was still a lot of, but the effects still held on day 8. If you have any more questions, I'm happy to speak with you after, but I have to finish, we have a time crunch, but I will definitely talk to you later if you want. Alright, so basically what this is saying is that the VLA EC pathway regulates acquisition of contextual fear. Alright. So, what does not regulate expression? So what about expression? Future studies should look at possible expression-mediating pathways, right? One might be the hippocampus to the amygdala, the hippocampus to the prefrontal cortex to the amygdala. There are a bunch of possibilities that these guys could look into to get an even fuller picture of contextual fear. And since contextual fear plays such a big role in PTSD, knowing as much as we can about contextual fear on a molecular level could eventually lead to an understanding of how PTSD works, and thus maybe more biological treatments instead of symptomatic treatments for PTSD. Yes?
I mean, in the future, would they try and maybe look at prevention of the acquisition of the PTSD? So, let's say in the case of a soldier, in that context, would they maybe work on that rather than on the expression? Like, after?
What you're talking about definitely would happen in all likelihood, but further down the road, because you can't really experiment with humans until you've done a lot of animal studies. And when you're messing around with humans' brains, you want to have a very good picture of what contextual fear looks like in mice. And to do that, we need the expression half of it as well as the acquisition. Any other questions? Yes,
Are there any other pathways that kind of like scientists know that help with PTSD, or is this like kind of like one of the?
I only got half of that. Sorry.
I'm saying that are there any other pathways that scientists know that might help with PTSD, or is this kind of?
This is the only one they discussed. As a general rule, hippocampus, amygdala, prefrontal cortex. Odds are a combination of those three in some way will affect fear. Very broadly speaking, this meeting. Okay, so, thank you. Especially me, Dr. Lucille, Mr. Maxwell, Hero Club, thank you for helping me put this together. Claire, Sydney, Catherine, Amanda. Thank you to IRT and to all of you. Any other questions? Yes, Mr. Goody.
Okay, so if you're acquiring a fear and that's intact, then the memory of the fear as well as the emotion associated. So like, lightning and healing sphere
Yes.
are stored together.
Yes.
But if you just acquired one without the other, then let's say you would acquire memory of lightning, but not the fear associated with it.
Yes. And I'm not scared when I see lightning because I'm never scared of thunder. So I know what lightning is, but I'm not scared of it.
So you would, every time you experience lightning and thunder, you would be afraid. However, when you access the lightning memory, you would not also access the fear aspect.
No, if you've already acquired fear, let's say you are afraid of thunder,
Right.
and you're afraid of lightning too, or associated too.
Right.
If you pull up lightning by itself, yes, you are going to be afraid. Because
What I'm saying is if they inhibit the
happen.
recall pathway,
Yeah. The recall pathway, okay.
then you would pull up the image of
Yes,
lightning
yeah, okay,
without
yes.
the fear. So you would know lightning and thunder were associated, but you would not be afraid
Yeah.
of either.
Yeah, probably a little bit, but not as much, yes. Yes.
What would be the freezing of mice who haven't had any BLAEC inhibition?
They would freeze when they were scared. This would be that. These are control mice. This is how much they froze without any inhibition at all. So these are those levels, right? You can see there's a lot less of those bars in this area right here because these guys got their pathway inhibited during acquisition.
But then what about is the expression one because what I'm trying to say is there's two graphs, so the left one's the inhibition during acquisition.
And it's no control. Yeah, the expression one would look like this. Or at least very close to it.
Okay.
Alright. Any other? Alright, we gotta go.